DMG informational webinar as part of National Brain Tumor Awareness Month here in the US. My name is Bruce Campbell, co-founder of the PNOC Foundation, whose mission it is to raise funding, resources, and awareness in support of PNOC's critical research and clinical trial efforts. Today, you'll hear some of the latest thinking on research and clinical trials for DMG from our esteemed international panel of leading experts working passionately and tirelessly to improve outcomes for kids and their families diagnosed with DMG. Full bios for our speakers today can be found in the chat. As always, we encourage you to ask questions, but please do so using the Q&A button at the bottom of the screen in the circle with the 3 dots and not the chat. Also, gentle reminder that this is not a forum for individual diagnosis or treatment discussions. Second opinions or consultations can be arranged by reaching out to your treating oncologist and asking them to in turn reach out to any of our panelists or PNOC investigators. To lead us off, please welcome PNOC co-founder and UCSF Professor Emeritus, Dr. Michael Prados.
Hi, and welcome to everybody. This is going to be a very exciting webinar. I am certainly not Sabine Mueller, and she apologizes that she can't be on because of jury duty today. Life seems to have changed a little bit for her today. But we have an incredible panel. We're going to talk about some of the opportunities that we have in trying to deal with this group of diseases called diffuse midline glioma. And I think each of the panelists will talk about some aspect of it that we're very excited about, that forms a story that will help inform how we develop clinical trials. So we'll start out with Jasper, who will be talking about an exciting new strategy using an oncolytic virus. And then we'll move towards metabolomics and other biomarkers, and then imaging to round it off at the end. So please feel free to ask questions. We'll get to them at the end, and we'll try and answer as many as we can. So with no further ado, we'll start out with Jasper to talk. So go ahead, Jasper.
Yeah, so thank you very much, Mike, and thank you very much for letting me giving this webinar. I'm very excited to do so, and hopefully I can make myself clear and the enthusiasm I have for this new revenue of treatment. Indeed, feel free to ask questions now or later on. So, as said, I'm going to talk about oncolytic virus's potential treatment for diffuse midline glioma. And then I try to go to an ES. So a bit immunotherapy of CNS tumors. I think there's quite some action going on around immunotherapy within DMGs as well. Also CAR-Ts, but I will not talk about that today. I think we're all aware about the poor prognosis that diffuse midline gliomas have. And to make immunotherapy more workable, I think an important task is here to change the tumor microenvironment from a cold towards a hot microenvironment, meaning that your immune cells that are now within diffuse midline glioma not very active and even helping the tumor to protect them, to protect the tumor to the immune system, to make them more active so that T cells and immunotherapy can work better. So we need to look how does the tumor microenvironment looks, especially when we give immunotherapy treatment. So to a bit more simple, all these different pictograms are all different immune cells, and they are kept away from the brain, as you can see when there is a tumor. So I don't know if you see my arrow, but you have myeloid cells that are very protective towards the tumors. You have kind of receptors on your tumor cells that prevent T cells from recognizing the tumor. So all this we need to circumvent. And what's happening is that within the prison, so to say, there you have the tumor cells. You have to find smart ways to get into the prison to attack the tumor. So now this wall we need to break down. The viruses, what does it help us? And I'm going to talk about the adenovirus. And the adenovirus is a DNA virus, which we know to cause common colds. So a lot of people has gone through a common cold that has been caused by the adenovirus. But now we are modifying by a company called CV Bio, has been modified the virus on 2 different spots, and I don't get into detail, but this modifications make the virus more can enter the tumor cells more easily, and also it cannot replicate in healthy cells. So of course that is very important, that you don't get sick or other cells get infected. And so what actually is happening then, that you get an infection of normal cells in the green, and you get an infection of the cancer cells. And as you can see, the replication stops once it gets infected due to this modification, and keep on replicating in cancer cells. So a bit more detail. So I think it's important to stretch that there are 2 mode of mechanism of action. So first, the virus infects selectively the tumor cells, as you can see under 2, and then do the replication. You have the oncolysis. That's why the name is oncolytic virus. And by this oncolysis, you get a lot of proteins that are released from the tumor cells. And these proteins are recognized by your immune system. And this immune system starts to recognize also other tumor cells, and ideally attacks all the tumor, and you build up also memory against tumor cells. So these 2 mode of actions are very important, and more we think maybe the second one, so the recognition of the tumor of the immune system, is even more of importance. So here I like also always this kind of cartoon thingies. So here you see the oncolytic virus has infected the cancer cells, and now this T cells say, hey, looks like a serious threat. So you get the immune system that is now going after the other cancer cells as well. So this is a kind of simplified mechanism of action to understand how this actually works. We always give this virus, or in the trials that has been done so far in adults, and also one what I will explain now, are injected directly into the tumor. So therefore, you don't get the antibodies to neutralize the virus directly, and of course, you get more directly into the brain, causing inflammation that you want. So you first make an MRI to really make sure how you get towards the tumor, and secondly, then with a channel, you go to the trajectory of the biopsy. You move your catheter a bit, and along the catheter you give the virus. And as you see on the MRI images, you see the gadolinium, which is also administered through this channel. You see a whitish area, and there is where the virus is delivered. And so you don't need like convection enhanced delivery like with normal drugs you can give also directly into the tumor. You want to enhance as much as possible, but here ideally the virus replicates, and then the immune system does the second hit. So a lot, and most of the work has been done preclinically by Marta Alonso, and Jaime, her colleague, is leading the clinical part of this trial that has already been completed some time ago, and maybe people are aware of this trial that has been published already some years ago, where 12 DIPG patients were included. to see what is the safe dose for this virus. So they tested 2 doses, and pretty easily they could go to the highest level. And in combination with radiotherapy, they gave 1 time this virus. So what we have seen there is that you got a median overall survival of 70.8 months for this group of patients. Not all had the mutations, but the big majority had. You saw 1 patient who also had a good response of the virus, but taking into account he also got radiotherapy. That's also difficult to really see what's what. But this patient survived for over 5 years, and there was also longer-term survival, but it was not officially a DMG. So it was safe, and we saw some encouraging survival data. We thought, how can we further improve this treatment or utilize this treatment further? So we came up with the next trial, which just recently opened and also already included the first patients, that we repetitively infuse this virus. This is called Delta 24 RTG. We repetitively infuse those patients also after radiotherapy instead of before radiotherapy, as has been done in a former trial. And it is really based on the field. I mean, the field is moving towards having viruses repetitively infused. And one of the most important studies that has been done in adults for glioblastoma has been done by a herpes virus, which was repetitively infused. And in recurrent glioblastomas, and there they saw very encouraging survival date of 28.8 months, which is long for glioblastoma relapse. And it was based on this trial. It was approved. I must say the numbers were limited, but also here the data was encouraging. And why is this field moving towards repetitive infusion? That you want to enhance and unleash this immune effect that I was talking about earlier. Is it safe to have repetitive infusions? PNOC already has completed a trial with a compound, so not with an immunotherapy compound, but with the panobinostat, which was given through convection-enhanced delivery multiple times in patients. And I won't go into detail of this trial, but the message here that you must take along is that here patients has been received multiple infusions of these drugs in the brainstem where the tumor was, and there were no big safety concerns there. So therefore we feel that repetitive infusion can be safe. So this trial is really to look at how much virus administrations can we give safely. And so we start first with 2. If that is considered safe, we move to 3, etc., with a maximum of 6. And the patients are enrolled after radiotherapy, as said, and then the virus is administered, and then as the third time, we also do again a biopsy. And that is, of course, very valuable information that we really have on treatment information, which is pretty unique, as we mostly have either autopsy material or diagnosis material. So this will provide us with a lot of information, and with this information, so this is the design. So we have, because you can imagine that the tumor that is in the brainstem or somewhat higher in the thalamic region have different risks when it comes to administration. So therefore we have 2 different groups, the pontine and the thalamic, and we will look individually to these cohorts what is safe in that perspective. And so I come back to the picture that we showed earlier, as we have now the opportunity to see, are we with this virus, for example, changing the tumor microenvironment from a cold towards a hot microenvironment, and with these different time points of biopsy, we hope to answer this question for this particular compound. And I have this cartoon of children standing on each other's shoulders, because I think that is always how we work, and the patients who are participating on this trial are the next ones to give information, so that the children standing on the shoulders of the former generations of patients with a DMG to help us little steps further. So I hope it was not too quick or too slow. And thank you for listening.
Thank you, Jasper. I think we should move on, and Raul is going to give us a little bit of knowledge about more of a macro involvement with metabolomics that are an incredibly interesting field right now. So Raul, go ahead and start.
Perfect. Do you see my slides?
Yes.
Okay, perfect. So my name is Raul. I'm a clinical epidemiologist, and well, actually, we stay a bit in the field of the microbiome, right? So the virus therapy, but now we are talking about a broader sense. And today I want to talk about understanding the role of the gut microbiome in pediatric brain tumor patients. But to kick off, I think it's also important to know what the microbiome is. And the definition of the microbiome is basically a community of tiny living organisms that live in and on our body. And this contains mostly bacteria, but it also includes fungi and viruses. And this whole ecosystem is called the microbiome. And in and on our body, we have different compartments. So we have a skin microbiome, an oral microbiome, but also a gut microbiome. And the gut microbiome is interesting because it's the most diverse microbiome that we have. It contains trillions of different microbes, but they also play an important role in our health. They help us with our digestion, what is quite logical, of course, but they also play an important role in our immune system. And by producing metabolites, they contribute to our overall health. And when this microbiome is disrupted, we call it a state of dysbiosis. And dysbiosis of the microbiome, and especially the gut microbiome, has been associated with a lot of different diseases, like inflammatory diseases, brain disorder, but also cancer. And this association, this dysbiosis and these diseases is, in some cases, etiological, so they contribute to the occurrence of the disease. But for us, it was quite interesting to look also on how people respond differently to treatment. And in the cancer field, this basically started in 2018. And this is quite a notable paper published in Science, a really respectable journal. And the authors of the paper looked at melanoma patients who received immunotherapy. And immunotherapy has quite a diverse response rate. Some patients do respond, others do not. And they wanted to see if there was an association with the gut microbiome. So what they found and reported was that patients who responded had a different gut microbiome than patients who did not respond. And this is important because the gut microbiome can then maybe explain why some patients benefited more from the immunotherapy than the other patients. So the key message here is that it was the first paper that really showed that it was not just important for digestion, but it really affects the immune system and influence how well some cancer treatments work. A lot of other studies have validated these results, but then the next question, of course, is, can we then influence the gut microbiome? And there are a lot of strategies that you can use, but one of the strategies is fecal microbiota transplantation, or short FMT. And this is a procedure where we transfer gut bacteria from healthy people or patients that responded to the therapy to patients who still need to receive the therapy. And the overall goal is to improve the gut microbiome in the sense that it will eventually lead to better treatment outcomes. And this is from a more recent study. They looked at, again, melanoma patients and also the same immunotherapy, but now they complemented it with the FMT. And they wanted to know, first, is it safe, of course, because it was a phase 1 study who mainly looked at the safety, but they also wanted to see whether changing the gut microbiome could help the immune system better respond to the cancer treatment. And you need to trust me because time is limited, but if you look at these lines, if they decrease, it's better. So blue is better. And overall, the outcomes were better. So some patients had tumor shrinkage or disease control, suggesting that the microbiome may be modifiable. And although this was an early phase 1 study in a small group of patients, it really showed that it was quite promising, but still we need larger trials to confirm these results. And this brings us to the CNS field, right? Because we now know it affects immunity, treatment response. Others have shown that the side effects of treatment are also related to the gut microbiome, and it could therefore help explain differences between patients. But it might also be an avenue for personalized care. So we built microbiome research into clinical trials, and we did this together with the great team of all the PNOC people, Simi Müller, but also with Jasper van der Lucht. And we have a lot of active clinical trials like the PNOC 022, 023, 029, where it's already embedded. And basically in all the new trials, we also collect the microbiome to learn as much as possible from the patients that are included in the clinical trials. And we also have a lot of planned studies still. But then to our first results, right? Because we already collected a lot of microbiome samples, and the PNOC 022 study we analyzed for the major part already. And if you look at this figure here, you see all kinds of different bars, and all these bars is one sample. And if you then look at the bigger bars below, you see the different cohorts. So this is before radiotherapy, this is after radiotherapy, and this is at the disease progression. These are all different patients, and all the visits longitudinally are below here. So a lot of different patients, but some patients also have multiple samples. And what is really notable and what is quite clear is that these gut microbiomes are highly variable between patients, but also possibly between the visits. This is quite interesting because how do we then analyze what the impact of the gut microbiome is? So therefore, we focused on overarching effects to link the bacteria to the treatment response. And if we zoom out a bit and we look at overarching effects, we use diversity as a first step. And the diversity tells us how rich and varied the gut bacterial community is. We have two important measures there: alpha diversity. So if you explain it simply, you basically say how many different bacteria are present within one sample. And the beta diversity tells us how different one sample is from another. And if you look at these bars, this is one alpha diversity metric. You see that it's pretty stable over time. These are the different cohorts, and it looks pretty comparable. The only thing is that there's a bit of an outlier that it shows here at the progression visit. It looks like it decreases over time. The same here. This is beta diversity, and the more the overlap, the more basically the outcome is the same, or the beta diversity is the same. And it all is pretty comparable with the exception in this cohort with the progression visit. So the overall shape of the microbiome community changed only modestly over time. And this is good for us because then we can also clearly link it to clinical outcomes. But we also want to know, are there certain bacteria that we can link to survival outcomes, of course? And we performed a special technique, which is metagenomics. Then we basically know the genes of the bacteria that are within a sample, but we also can overlay this, and then we know what these bacteria are doing. And we call this gut microbial functions. So not just bacterial names, but we also know what they do. And this is an example of cohort 2, and then you get a list. And we differentiated between patients that had a short-term survival and a long-term survival. And then you see which bacteria are more active in the long-term survivors and also which are more relevant for the short-term survivors. And we combined this for all the 3 cohorts. We repeated this, and survival outcomes were a bit different across the different cohorts. So we made new cutoffs for all the different cohorts. But then again, here also we found a consistent microbiome signal across all the different cohorts, across all the 3 cohorts. And what was quite fascinating is that the long-term survivors showed a more metabolically active gut microbiome. And short-term survivors showed features suggestive of gut dysbiosis. And like I said at the beginning, this has already been linked to different disease states, but also to treatment response. And we specifically found an oral-like microbiome signature, and that is not what you would expect in a healthy gut microbiome. We found some bacteria or families of bacteria that are really important in determining the difference between the 2 groups, like the actinobacteria, the firmicutes, and the Bacteroideota. But for me, the interesting part here was especially that long-term survivors had higher enrichment of pathways linked to the short-chain fatty acid and tryptophan-derived metabolite production, which basically are signs of a healthy microbiome. We also tried to link the gut microbiome to toxicity. And this was an example of a severe treatment-related skin rash. And some of the patients in the clinical trial got this, and it was very hard to treat, and other patients did not got it. So we really could leverage the difference between the 2 groups. And what we observed was that patients that not had this severe skin toxicity, we found more commensal gut functions, enrichment of a specific bacteria, Prausnitzi, and also the related pathways. And again, we found that the short-chain fatty acid production was an important aspect here. And then also we found patients with a severe skin toxicity that were having more B vitamin-related pathways and more sulfur amino acid metabolism. And this might be an indication of that the gut microbiome can influence skin inflammation through the gut-skin immune axis. But then again, these findings are exploratory, and we really need to validate this with additional analyses like metabolomics that we can do on the feces, but also on the blood, and also immune profiling is an important aspect here. In conclusion, our PNOC 22 results showed that the microbiome of patients with the DMG was relatively stable, and that's not what we would expect with all the treatment they received. It was quite interesting. Also, the lower diversity could be seen as progression, but it's really something we need to investigate more. And we really found that microbial features were associated with survival outcomes, but also with toxicity. And this is an important first step for us because it helps us understand differences in treatment response, but also the side effects. And more importantly, it may guide future strategies to enhance the efficacy and reduce toxicity of these treatments. So this is really the first step. And we could not have done this work with all the support of these great foundations. And I want to specifically thank the Gold family for their support of our research. Thank you.
Thank you, Raul. That is amazing. There's so much more to learn here. It's great to have this repository of gut samples, right, from our patients to kind of correlate as a biomarker, but maybe as an interventional strategy as well. Our next talk will probably lend a little bit into the biomarker field. You know, how is it that we can establish what we're looking at is real, what's ground truth? And Benjamin is the perfect person to tell us about that. Benjamin?
Unmute myself first. Thank you, Mike. I'm just going to share my screen here. Thank you all for the opportunity to speak today. Can you see my screen okay?
Yep.
Great. So today I'm going to be talking about bringing AI to response assessment for DMG, kind of the state of where we are and where we'd like to go. I'm an associate professor at Harvard Medical School with an imaging lab focused on these techniques. So there's several fundamental challenges for response assessment for DMG as well as other pediatric brain tumors, and that is that we largely base this historically on practicality and what is feasible to do given clinical volume in the radiology clinic and in our oncology clinics. And so the current standard is to really do a two-dimensional measurement, cross-sectional measurement of tumor in the plane that has the longest axis that can be identified. And so there's three main planes that are looked at: axial, sagittal, and coronal. And what you can see is if you have a tumor that's like this, that's a little more diffuse, someone could rationally measure this in three completely different ways that would yield three different cross-sectional areas. And beyond that, that's only in one plane. We also, as I alluded to, have these three different planes. So here you see an axial plane measurement and a sagittal plane measurement. And when we're looking at response assessment to a certain treatment, such as radiation or maybe an experimental therapeutic, if you measure these tumors in these different planes, you can get quite different measurement. You can get quite different changes in measurements, which could even get to the point where you might have different interpretations of response of the tumor. And we see this in clinic, we see this in clinical trials, and essentially it becomes a problem because we cannot react to what we cannot measure accurately. So I think that AI can enable better, rapid, accurate 3D characterization of tumor response. Ultimately, what this enables us to do is have a rapid auto segmentation of the tumor in 3 dimensions. Now, one could attempt to do this manually, but it's just overly burdensome and time consuming, and just doesn't happen in reality. So with these newer AI tools, we can get a segmentation of the tumor. We can look at different subcompartments of tumor, and then we can calculate, and we can actually visualize differences in 3 dimensions. And you can see for many tumors, they're not shrinking and growing spherically or kind of in a circular manner. So we're just able to get a lot more granular assessment of what's going on with the tumor. And then you can create kind of these maps that look at subtle changes, distinct changes that have spatial accuracy and geographic importance potentially. As I mentioned before, this also enables us to look at different subcompartments like cystic components, edema, enhancing tumor, and gives us a practical way to monitor these over time longitudinally, where you could get these readouts of what's going on with the tumor volumetrics. This is a study where we applied one of the current state-of-the-art algorithms to the PNOC 008 and 022 data sets to look at comparisons between how radiologists were measuring tumors versus 2 different AI techniques, one where we had 3-dimensional volumetrics that were being done, and another where we had an AI-based 2-dimensional assessment. And what we found, these are just 2 examples, but there were some cases that were more concordant than others, but there were some where we had significant disparities where we actually did have the AI output, differences in response designation where, in this case, the AI output progression where the radiologist said partial response. And so, I think it really, this data is under review for publication, but what we're seeing is there are discrepancies, and this just brings up that I think we need to do a better job at kind of standardizing response assessment and using all the information that we have in a quantitative way. To develop these models, one of the huge challenges and opportunities is curating enough data to help us do these things and develop these models. So we have a collaboration internationally that's based at Dana-Farber, Boston Children's, but also with UCSF, Pediatric Neuro-Oncology Consortium, University of Michigan, and ETH Zurich. And so we've curated a data set of 834 patients and almost 5,000 scans, where we're going to try to annotate these to really help develop more accurate algorithms. Because despite the promise here, there's still a lot of work to be done. There is a number of groups working on developing better models that can do more accurate segmentation. But a real unmet need is to do this in the kind of post-treatment and longitudinal setting. So we've made some efforts this way also, developing a model that was specifically trained to perform better in the post-treatment setting, and just showing you some of that data here. This DMG tracker that we have had a higher acceptable segmentation, acceptable segmentation accuracy compared to the prior state-of-the-art model. But one thing to note is that we're still only at about 65-ish% in terms of putting this model through and getting an acceptable volumetric segmentation out of the box. So I think there's still a lot of work to be done. Another interesting area for applying AI to imaging for pediatric brain tumors, including DMG, is to try to develop kind of these digital or imaging-based biomarkers that are detecting subtle changes in signal intensity over time that then might predict the future state. So this is a model that we had developed that analyzes longitudinal imaging for pediatric gliomas. And then you can basically feed the algorithm sequential scans, and it will make a prediction about what will happen within the next year. Is there going to be recurrence or relapse within a year or so? And I think that there's a lot of promise in developing tools that kind of function in this way as well. Taking this even a step further, there are opportunities not only to try to predict will relapse occur or not, but actually predicting, using prior information from an image as well as things like the radiology report historically to actually make a prediction of what the tumor will look like on the next scan. So what the scan itself will look like. With all the advances, even over the last year or so in these generative models, we now have the technology to do this. But these models still, there's a tremendous amount of work to be done to actually make sure these models are accurate and reliable, particularly in the setting of pediatric brain tumors. But all in all, I think the vision is that we're going to go from kind of this subjective interpretation of what we see on the left to having information that we're able to extract quantitatively on the right, and longitudinal information about tumor size and trajectory, and other predictive biomarkers potentially as well. I just, with the last couple of minutes, wanted to just highlight some specific challenges and opportunities in this space. So one, probably the biggest challenge is that DMG is incredibly rare, so around 300 cases in the US per year. For good reason. And however, from an algorithmic standpoint, this is challenging to create algorithms that are going to perform robustly in this setting. So there's opportunity to pool data, build these federated imaging commons, and we're trying to work on that as well as other groups. We also need a lot of support for expert annotations. So these are these kind of ground truth tumor segmentations that we can get en masse to help train these algorithms, and also a lot of work on harmonizing how we acquire these images across different institutions. Another challenge, is that simply the models we have right now, and using this kind of two-dimensional representation is not precise enough to really give us a granular assessment of response. The models we have also, as I showed currently, still have pitfalls. And so we need to keep working on model improvements, validation studies, prospective testing of these within cooperative group DMG trials. And also something that I'm not directly involved in, but I know a lot of other groups are, is working on advanced imaging parameters like diffusion, perfusion, and other kind of molecularly labeled imaging tools. Another is that we don't have any current imaging predictive biomarkers for DMG. And because of this, in part, we treat every child more or less the same because we don't have a real way to personalize based on these trajectories of what's happening. So I think a lot of work into developing these models, as well as very importantly here, is linking, creating databases and repositories that link imaging and genomics, including tissue data and liquid biopsies, to really help inform this more personalized approach. I think finally here is that a lot of these models we develop at just a few centralized institutions, and we have to make sure that these algorithms are going to work more robustly nationally and also internationally on different types of scanners that have used different protocols. So I think there's some opportunities to help unify the way we process these images before putting them into the algorithms, as well as cloud-related delivery tools to help create tools that we can really use everywhere. And this also requires a lot of validation as well. And then finally, I just wanted to mention we are working on a lot of these issues actively within the context of this, what we call the AI RAPNO initiative, which is the mission of this group is to promote safe, effective, and clinically meaningful translation of AI tools for response assessment in pediatric neuro-oncology and ensure that these enhance clinical expertise for these patients. And Dr. Brunig, who you'll hear from next, is also on the leadership team for this initiative. So in summary, I think, there's potential for AI as we apply this to DMG imaging to better understand tumor biology and trajectory, to have more precise measurements of response, to promote consistency and standardization across readers and centers, ultimately help design smarter clinical trials where we can use kind of these personalized outcomes and predictive biomarkers to help see who is eligible and who would be best eligible for certain trials of intensification of therapy. And then ultimately have a foundation for personalized management that could direct therapeutic action. So thank you all very much, and I'd be happy to take questions at the end.
Thanks, Ben. That's amazing. This is going to be such a huge body of work when it finally comes together that it'll help us sitting down in front of a patient, looking at a scan and trying to understand truly what it is that we're seeing and how therapy is influencing that. Which brings us to Sarah, who's going to round this all out, right? You're going to talk about how to use all of these tools to help us in the clinic. So Sarah, go ahead.
Thank you very much for the kind introduction. I will quickly share my screens. All right. Yes, I want to continue where Professor Khan left off and see how we can actually move from an individual prediction and a segmentation on to using predictions in space and time on imaging for diffuse midline glioma patients. And I want to present this in the context of radiotherapy. Radiotherapy is really one of the major treatment options for diffuse midline glioma patients. Even though it is not curative, it still plays a major role in the context of the treatments of these patients. And I'm outlining you here the workflow of a radiotherapy treatment pipeline that starts from the initial consultation, then there is imaging taken, and importantly, afterwards, the tumor itself is contoured, as we just heard from Professor Khan, but also the adjacent healthy structures as well, so that we can calculate an individual plan for each of our patients. There's then some quality assurance done, and then this plan is being delivered in several treatment fractions. Typically, this is 30 independent fractions over the time course of 6 weeks, and we follow up thereafter. And I'm particularly leading here a project in this context, which aims to personalize radiotherapy even beyond the calculation of an individual plan. So we actually could also optimize the specific maximum dose that we treat these patients to, the temporal delivery of these treatments, and finally also the targets that we outline. So while we outline, and I will mention this a little bit in the next step as well, of course, what we see on the images, we also know that for diffuse midline glioma, actually the tumor also infiltrates beyond what we can see on the image. And therefore, it is very important to have an understanding of where this microscopic infiltration really goes to, so that we can include this in the target volumes as well. And then we want to use AI in order to facilitate this personalization. What we want to do here is actually to, first of all, use computational tools to explore if there is even potential for an improved outcome for our patients through the changes in the fractionation or the dose distribution. So what we need at first is a typical prediction model where we provide a prediction of the response of a patient as a function of time. And once we have such a model established, we can then change parts of the input. For example, we can change the spatial distribution of the dose and observe if this has an influence on the trajectory that we predict for this particular patient. So that is the goal that we would like to achieve. And the question is now, how do we actually get there? And we've heard already a lot about magnetic resonance imaging. It is really the cornerstone of the diagnosis and the clinical assessment of the patients, also in terms of the treatment decisions. and then of the response assessment, as we just saw in the previous talk as well. So what we try to achieve is to provide actually prediction in the space of the MRI so that we can make a better assignment of these target volumes that we see here outlined again, where this clinical target volume, the CTV, is really also encompassing this microscopic tumor volume. But secondly, if we have a prediction of how a patient would respond either without treatment or just with a standard of care radiotherapy, we could also use this for an individualized control to see if there's any differences compared to how a patient actually responds. I need to stress that this is definitely still research. This is nothing that we currently already can deploy in the clinic, but I want to give you an update on where we stand with these types of models. And we already saw the essence here is the data. And I wanted to also give you a perspective from a different center. So we are here located in Switzerland, in Europe. And of course, in order to be able to work with data from diffuse midline glioma patients, we need to pull this information from many international institutions. Well, there's a lot of legal requirements that have to be put in place first, because of course we have strict data privacy policies in order to facilitate this. Then even once everyone has agreed, we still need to facilitate a safe transfer of this information to, in our case, a central location. And then starts the part where actually we spend most of our time, and that is the preparation of the data and the cleaning of it. In this case now for the radiotherapy response, we also wanted to spend some time in terms of understanding how we define the efficacy in the context of a longitudinally changing system. And then finally, once all of this is prepared, that we have the data in place, we have our response label in place, then we can actually move on to train our model and evaluate the performance, which actually almost is the easy task thereafter, because technically there are really some great achievements already being done in this domain. So in this context, we also actually have a few side products that support us in this workflow. This is, for example, we're handling a large amount of individual scans, and we've designed a specific model that actually predicts how well a segmentation is going to be performed in order to prioritize which scans actually need to be manually corrected for these segmentations. Secondly, we established a separate model for describing the growth of these tumors, and also approaches how we can handle missing data, because a lot of the data is actually scattered. And in order to still use it effectively, we need to have specific strategies of how we can actually account for this missingness so that we can still use the large majority of these data. And so with this, I also wanted to summarize actually a very similar cohort to the one that we just saw from Professor Khan. This is really a great effort to centralize data from international institutions across continents here, and also harnessing, of course, the great resources from the clinical trials, so from PNOC and also the CBTN, so the Children's Brain Tumor Network, in this context. And since we're talking about longitudinal imaging, I also wanted to give you an overview of, we have almost 4,000, we have over 4,500 individual sessions included here, but it varies a lot for each of these data sets, how much information we actually have available. And this also means that for each specific project, depending on what information you want to have, we have only a smaller subset of the data that we can actually use. So for example, in our case here, we have around 200 of this larger pool of patients that we can actually use for the radiotherapy response modeling. So with this in mind, I also wanted to mention a few of the key limitations, which is first of all, that we are handling here multi-institutional data, also covering a large time frame. So we have variation in terms of the scanners, and therefore also variations in terms of how these images are represented. We have a lot of missingness on all sides in terms of individual contrasts, in terms of certain time points, in terms of metadata that we are, of course, now trying to really piece together with an effort that really involves a large number of teams across all of our institutions. So with this, I want to also show a few of the examples that we have already achieved. The first thing I want to highlight is actually not an AI model, but this is a math model, where we explicitly describe how a tumor responds to radiotherapy and how it will regrow thereafter. So in blue, you can see the individual data points, which is tumor volume assessments from a three-dimensional segmentation that we saw before. And then in red, we fitted the data with this mechanistic math model. And then we provide a prediction for a later time point where you can see that at a later time on the right, you have this interval going up because, of course, there's larger uncertainty if we project further into the future. But we can still see that we actually capture the relative trajectory of these tumors already quite well now. And as I said in the beginning, the first thing is to understand, and now we can actually play with this model to see how this trajectory could potentially be influenced if we change aspects of the model that describes our radiotherapy response. Secondly, we've also made some good progress in terms of predicting the individual radiotherapy response that we quantified based on the longitudinal trajectory of these patients. This quite or it is not that rare that actually we have first a progression that seems like a progression. It's a so-called pseudo-progression. That is not an actual progression, it's just something that appears like it on the imaging. And so therefore, we really wanted to have all the information of response assessments available to build us more robust models that we could actually then also predict in this context. And lastly, I want to talk about the image generation. We saw one example already from Professor Khan. We actually use a slightly different approach using so-called denoising diffusion models. You might have used these actually yourself already if you generated images, for example, with ChatGPT. We use the same concept, just that we give one MRI as an input, and then we predict this MRI at a later time point, using this math model I showed you previously in order to guide us, so we can actually provide a representation of a particular image slice at this later time point. And we've actually now applied this based on the data that we have. And it's really interesting that we have some promising results so far already that we can provide tumor growth probability maps from a starting image. So not just one image, but actually we can also repeat this process so that we have these tumor growth probability distributions. And it was really nice to see that our radiologists were actually unable to discern a true from a generated image, and also the directionality of the growth that is predicted here very much agrees with what we have observed. But of course, this is just the start, as I already mentioned. So we continue to establish the structured database, which is really the foundation of all of the AI predictions that we're making using in this context. We have a model that provides really good first results, but we of course want to validate this now further and also move to more realistic representations, also in counting different time points in the time course of these treatments. And then of course, coming back to continue our goal of providing individualized predictions in terms of response to radiotherapy and also investigate alternative treatment scenarios. So with this, I would like to thank you for your attention. And I do want to also emphasize that this is really not just an effort by myself, but by a whole team, a lot of which are on this call actually. And it's really great to join together in order to improve this. Thank you very much.
Thank you, Sarah. There's a lot of great questions coming up, and I'm not sure we can get to all of them, but I'll try. I want to stick to I want to start with the immunology a little bit, and Jasper, maybe you can help with this. There are a lot of strategies: CAR T cells, different oncolytic viruses. We've done measles. You're talking about adenovirus. We talked about the herpes virus, and then also how the gut is going to influence perhaps the innate system or the adaptive system of the immune system in children with brain cancer. So maybe just very briefly, kind of think about how you prioritize what you think would be the highest, or the priority. What do you think would be the best priority to start moving forward in children with DMG, knowing that all of these opportunities exist and they're expensive. All of them are expensive to do. I know that's a big question, but Jasper, do you have any thoughts about that?
Yeah, sorry, Mike. I was kicked out of the meeting, so I'm just in. So I only heard the last part of your question. So
The question, I guess the question is, there are a lot of potential strategies to try and harness the immune system in children with brain cancer.
Yeah.
How do you prioritize, and how do you think about which should be at the top of the list of things to do?
Yeah, so of course that's a very difficult question. To me, for example, checkpoint inhibition is something alone that may not be enough because it doesn't enter the brain, etc. But I think hopefully this trial with repetitive infusion and on treatment biopsy can learn us, okay, what has been changed in the microenvironment and what should be added to make it more effective. So if you see an upregulation or a change in your, say, innate immune system, so then you see, okay, there has something changed. But if you still see, for example, exhausted T cells, there maybe you need another compound. So I think we really need biological information that can help us to decide on the next thing. And I don't believe there will be one compound, one CAR T, one oncolytic virus that will do the job on itself. So all those phase 1 trials or phase 2 trials ideally help to give a piece of the puzzle, but then we need to see what are smart combinations. And even then, it doesn't necessarily have to be immunotherapy with immunotherapy. You can also say, okay, do you need a small molecule to upregulate to change your microenvironment first and then add an immunotherapy compound. So I believe we need to go towards combinations, and with really assessing our patients so in depthly will help us to make these decisions better.
Yeah, I agree. It's a complicated question, and money always plays a role, right? How can we even do these kinds of things? I wonder if Ben and Sarah could comment briefly about predictive biomarkers using AI and imaging, and if you ever envision we have an opportunity that we can actually make changes before we see things, therapeutic changes before we see things on a scan, because that ultimately probably is the best way to deal with the burden of disease, and it gets again to the ground truth. What's actually happening in the brain, and can we understand it?
Sure, I could jump in, or Sarah. I think that that's a really great application of this, and I think we kind of both showed examples of models that I think in theory could do this, where if you had a few scans of a patient, or I think in Sarah's models maybe just one scan, you predict what the future state will look like, and then if it meets some criteria, if it's showing growth or some significant amount of growth, that would be, you could initiate treatment really before even getting that scan. So I think theoretically the technology is there, and I think the question is now I think we still need to validate this, in kind of a prospective setting, or at least kind of like a silent prospective setting where we have like a simulated study where we see how the model would have like performed on a specific trial. Because I think we still have to do some investigation. I don't think that, for accuracy's sake, to make sure the model's not picking up kind of spurious signals based on the data it was trained on, and it will actually work out in the real world.
And maybe I will add one point. I think it's also not just about making a prediction of change in response, but this could also influence, for example, a follow-up schedule. What we saw now in the data is usually, at least for diffuse midline glioma, there is often one trend, but if regrowth occurs, then it's a very fast growth. And in that regard, to have a probability of when it would be a higher risk that potentially at the next scan there would be progression for a patient to come back a bit earlier, that this could be captured at a very early stage. I think this would also be good, because it's not about, I think we're far from relying only on an AI prediction to change the treatment. I think we're not there. So it would probably be a first step to ask for an earlier follow-up, and then react still based on what is observed actually in the patient.
There's so many other things to talk about, but we've run out of time. I think I'm going to turn it back over to Bruce to end this. But I guess the message that I would like to say is that we have to continue to share data, and that we have to engage our patients and families as partners, as real partners with us. And the more prospective trials that we can do and how we can put this data together in a rapid way will just move the field forward so much faster, and we don't have time to waste. So Bruce, I'm going to turn it back over to you.
Sure. Thank you, Dr. Prados. Thanks to all of our panelists today. Really grateful, invaluable information, and stepping away from incredibly busy schedules to actually share this with the audience. We managed to actually answer 7 of 10 questions in the Q&A. We've got 3 open. We'll attempt to get those on the other side of the call. So stand by, and then we will circulate a full recording of today's webinar to all of the registered participants. Thanks again. As Mike said, it really takes not just the data sharing and the collaborative scientific effort, but a collaborative philanthropic effort as well. And special shout out to the Neve Colty Foundation and Misha for their pioneering work in the microbiome space and funding that important research. Thanks everybody for joining, and we'll see you on the next one.